Hello everyone. Welcome to another session of our COVID classes. In the last class, we had a general idea regarding mastitis and its etiology and how various etiologies manifest in a dairy farm. So today we will be discussing about diagnosis and, if time permits, about the treatment and various treatment strategies followed for controlling or for treating bovine mastitis. So let us see how we can diagnose mastitis in a farm. So when it comes to clinical mastitis, it is quite evident that and you can diagnose mastitis just by physical examination. And for further understanding the etiology, in an ideal situation, you will go for a culture. You will do the culture and identify the bacteria and do an antibiotic sensitivity test, and thus come into decision regarding the treatment of the particular case of mastitis. A good indication regarding a confirmed diagnosis of clinical mastitis can be a negative answer to the question that if the milk of the particular cow in question is drinkable. So if it is not drinkable, then definitely it is clinical mastitis. But things are quite different when it comes to subclinical mastitis. See, subclinical mastitis means the milk will be clinically normal. So if you look at the milk and ask yourself a question that if you drink the milk, definitely you won't find any problem with the milk on a cursory examination, or even with the cooking of the milk, or even during the consumption. But what happens is that large number of cows in the farm may be affected with clinical mastitis, and these animals may be having a suboptimal production. Thus, subclinical mastitis has a major impact in the dairy sector. And for this mere clinical examination won't suffice. Here you have to go for testing of the milk to see if the cow is affected. The ideal test to do is, of course, to collect the milk and do a cultural examination. And if you find that there is microbes in the milk, then definitely the animal is suffering from subclinical mastitis. But when you consider a large producing farm or a large organized farm, this is going to be a very expensive proposition. So what people do is that they go for an indirect test. So indirect test is relatively inexpensive, and most of these tests are cow side in nature. That means you can do this test, standing next to a cow, you will get the result immediately, or you can assess the result immediately, and then you can say if the cow is affected or not affected. So those are the peculiarities of indirect test. However, the indirect test, it detects inflammation but not infection. That means it detects usually detect if the milk is abnormal or not if the udder is abnormal, or if there are any bacteria or infectious organisms in there. It simply detects if there is any inflammation or if there is an inflammatory response to the udder. And though the sample, in an ideal situation, you collect the milk that are showing signs of inflammation, you send it for culture, and you will get the result in bacteriological examination. So that is how indirect tests are performed. Now, how do you detect mastitis at a herd level? So the general practice where industrialized farming is prevalent is to go for a bulk tank somatic cell count. So what they do is that they collect the milk into all the animals in the farm into a bulk tank, and then they do the somatic cell count testing and assess the number of somatic cells per ml of the pooled milk. And it is found that the bulk tank milk count, or BTM, or bulk milk tank somatic cell count, or bulk tank milk somatic cell count, BTM SCC, it is increased primarily by gram positive cocci, and that indicates contagious mastitis. So somatic cell count is in such countries or in such areas, the somatic cell count forms the basis for pricing of the milk. What we have in Kerala is that we price our milk based on the fat and SNF content. But in an industrialized country where the quality is very important, they fix the price of the milk based on the quality of the milk, that is the somatic cell count. And thus bulk tank milk somatic cell count or bulk milk tank somatic cell count is important in large number of countries, and that is the standard pricing method of milk. And thus if the somatic cell count increases beyond a limit, the price of the milk, they impose a penalty, and the price of the milk goes down, or the share of profit to the farmer is reduced. And another important feature is that if you know the BMT-SCC, then you can estimate the number of quarters affected, and increased BTM-SCC indicates an increased infection and an increased loss to the farm. So not only the price, or if there is a penalty regarding the price of the milk, but actually it directly indicates how much or indirectly estimates how much amount of milk is lost to the farmer. So they generally do this BTM-SCC regularly, and they fix the price of milk accordingly. So another thing to do is that so you can directly go for culturing the milk from the bulk tank. But the problem is that this can be reliable when it comes to, when you retrieve Staphylococcus aureus or Streptococcus agalactiae, but you cannot be sure if you are retrieving some other organ. Suppose you get E. coli in the milk, then generally you may get a coagulase negative Staphylococci. So these can be from other sources also. So the sensitivity is low, but you can say relatively high specificity is there, and there can be false positive cases because you may be isolating microbes from other sources also. Now, how can you detect mastitis at a cow level? So cow level mastitis can be detected based on a composite somatic cell count. That is, you pull the milk from all the 4 quarters and do the somatic cell count, and then you can go for if the cell count is high, you can go for culture and sensitivity. So such types of screening will help in indicate the prevalence of subclinical mastitis. Then you can the culture will give an idea regarding how the mastitis, or what is the mode of or how the cows are getting this subclinical mastitis. Suppose if you get Staphylococcus aureus, you can be fairly sure that it is because of contagious spread. If you are getting more number of Streptococcus agalactiae, it could be because of the environmental issues. So that is how you do. And then based on the isolates obtained, based on the number of animals infected, you can go formulate a plan accordingly regarding how to control or treat these cases. So that is how you do a somatic cell count. Then you can go for a culture at a cow level, and this has a very high level of sensitivity and specificity, and this is the most ideal thing to do. However, it's not very practical if you have hundreds of cow in a farm. So generally what we do is that we do only we do some indirect cow site test that I will be coming to shortly, and then you select the infected cow, select the appropriate quarter, and only those are cultured, and that can be really helping to decide on treatment. But again there are certain limitations, because the quality of your culture or the quality of your result will depend upon the quality of the sample collected. So if you are not properly collecting and processing the sample, you may get any other bacteria. See, even you can get bacteria that are present in your hand also. So the procedure of milk collection and processing of the milk for culture and sensitivity is very important. A cursory view regarding this, first you have to wash and dry the udder, then rub the udder with 70% ethanol. So you have to start from the tip and go towards the upper areas of the teeth. Then first 2-3 streams you have to discard, and then only you have to collect the milk. So that is known as the pre-milking sample. That means before you milk, before the cow is milked, you collect the sample. Then another sample is collected at the end of milking, that is known as the post milking sample, and these are collected onto a screw capped vials, and it is refrigerated. You carry it in a ice pack or a cool pack to the lab, and then you subject for culture and sensitivity. And if you are getting like 3 or more isolates, then the sample is considered to be contaminated, and the result is not used for a decision making regarding the treatment. Or how will you know that the udder is cured? To say that post treatment, again you have to do the culture to say that the udder is cured bacteriologically. Sometimes clinical cure may happen, that means the signs may go away and a cow may return to a normal level of production, but bacteria may still be remaining. So that is known as clinical cure, and there should be a bacteriological cure also. So how will you know that the bacteriological cure has happened? For that, after your treatment is over, you have to reculture the milk sample from the same udder at 14, 21 and 28 days, and the originally isolated bacteria should not be there. So if you have isolated Staph aureus, then after 14, 21 or 28 days, you should not get Staph aureus. Suppose you should not be getting any bacteria in the culture. Suppose you get during the clinical signs, or during when the milk was infected, you got Staph aureus, then after 14 days or 21 days you get Streptococcus agalactiae. That means the cow has acquired a new infection. So that is how you do a culture and sensitivity test at a cow level. And what if you do not get any culture in a case of bovine mastitis? So this happens. This occasionally happens in our labs also. So that can indicate many things. It could be because of a spontaneous cure, like as I probably told in the next class. Like in case of Streptococcus agalactiae, even E. coli, there is a chance of spontaneous cure. The body may react to the infection, and the infection is warded off. So the mastitis can cure spontaneously also. So it could be because of spontaneous cure, then could be because of a very low concentration of pathogens. Some pathogens may be, forming certain biofilms, or becoming intracellular forms or L forms, and they may shed into the milk only very occasionally. So if you are collecting milk when the bacteria is not shed in the milk, then again you may get a culture negative thing. Then there could be certain proteins, or there is this iron lactoferrin system that could inhibit bacterial growth. So it could be because of this certain inhibitors present in the milk. So these factors should be counted in when you are getting a negative result for your culture. So in such cases, what you have to do is that you have to go for a finer tests that are more sensitive, like you can go for ELISA or a PCR or multiplex PCR, where it will give even can detect even the presence of a very smaller number of organisms. But again, these tests could be, there is a high chance of being false positive, or these tests may have a very high sensitivity, but specificity of such tests can be really low. Then there are certain other tests which are not really that can be done but are not usually done. Like you can filter the milk, you can collect the milk, centrifuge it or pass it through a bacteriological filter, so that only the bacteria gets retained as the filtrate. You can wash it down onto a slide and you can stain it and see if you are getting any bacteria. So that can be done. Then there is another test called as a litmus test. So when you are doing this litmus test, you can detect the presence of endotoxins, and that is very important when it comes to coliform mastitis or infections caused by gram-negative bacteria. So detection of endotoxins is done by litmus test. So that is how you detect infections at a cow level. Now, this detection of subclinical mastitis heavily depends on various indirect tests, or indirect test is very important when it comes to detection of subclinical mastitis, as I told you, because the ease of doing, the relatively low expense, and the ease at which this test can be regularly done in a farm, so that the milk production can be monitored efficiently. So the first or the most important parameter to estimate in such test is of course somatic cell count, and the presence of somatic cell count or the increase in the somatic cell count directly indicates a infection. So the average somatic cell count normally expected in a good quality milk is about 100,000, that is 1 lakh cells per ml of milk. Now there is a particular formula where you can convert the number of somatic cells or the somatic cell count to a somatic cell score, that is scored from like 0 to 9 or 1 to 9, and based on that you can score the quality of milk. So that is how they fix the price of milk. So for 0 to 3 it will be a high quality milk, and as it tends towards 9 it tends to almost clinical mastitis. And a thing that you have to keep in mind is that the somatic cell count values will be very high in the early phase of lactation as well as in the later stages or towards the drying. So early and late lactation, the somatic cell count values will be very high, but the average somatic cell count. So when you are considering the somatic cell count of a cow, you have to factor in the stage of lactation also. And the average or during a normal lactation period, you can expect like between 1 to 2 lakhs cells per ml, and that is okay. California mastitis test is a very important cow side test that estimates the somatic cell count as well as the pH of the milk. So as I told you, I hope I told you in the last class that as in response to mastitis, there will be a large amount of serum components flowing into the milk along with this diapedesis of neutrophils. So during a case of mastitis, both the pH will increase as well as the somatic cell count will increase. So California mastitis test, which is also known as the rapid mastitis test, sham test, because Sham and Nulander were the persons who developed this test, or mastitis NK test. It estimates both the somatic cell count as well as the pH of the milk. So the normal somatic cell count is around 1 lakh, and the normal pH is around 6.6 to 6.8. And the peculiarity of this test is that it uses bromcresol purple as an indicator, and bromcresol purple becomes deeper purple in color when the pH of milk goes above 6.8. The advantage of somatic cell count test is that, the advantage of California mastitis test is that you can detect both pH as well as the somatic cell count, and you can get the result within 15 seconds. And how do you score the milk, the somatic cell count, how do you score the CMT, result of CMT? It is negative, 1, 2 or 3, and we will be learning more about this in our practical classes. So somatic cell count estimation by the California mastitis test is a very important cow side test. Another important test is electrical conductivity, which is very easy to perform. Only thing is that you need the particular equipment, electrical conductivity meter is needed. California mastitis test is still considered to be better test than electrical conductivity. So how do you do electrical conductivity? Electrical conductivity is a cow side test. And what happens is that as I told you, as the serum components in the milk increase, there will be an increased amount of sodium and chloride ions. So naturally, infected milk will have a higher conductivity. Industrialized, or if you have an industrialized milking parlor or automatic milking parlor, electrical conductivity is done by a robotic testing component will be there. Conductivity component will be there along with the milking machine, and they will be cow level conductivity will be recorded, and can even the peculiarity is that it actually measures the level of injury, because you will directly get to the amount of serum or the serum component that is present in the milk. And then another thing is the differential conductivity. So if you estimate, that is the differential conductivity means it is the difference between the highest resistance and the lowest resistance of the teat. So if one teat has a high or you get to the conductivity of individual teats, you subtract the conductivity, highest conductivity value with the lowest conductivity value, then you will get what is known as a differential conductivity, and that is that value is used for assessing the severity of inflammation of that udder. And the important thing is that this can detect mastitis very early. It can detect mastitis even before clots appear in Staphylococcus aureus or Streptococcus tuberculosis cases. So that is electrical conductivity. Then another mechanism is to assess the enzyme activity in the milk, and commonly assessed enzyme activity is in NAGase. So N-acetyl beta glucosaminidase is assessed, and this is actually an intracellular lysosomal enzyme, and you have to freeze the collect the milk, freeze it, and then thaw it so that the milk in the cells in the milk is lysed, and the enzyme is released into the milk, and these can be automated. Okay, these are automatable tests, and somatic cell count and NAGase tests are considered to be complementary to each other. So it is as good as assessing somatic cell count. Then other enzymes like lactose dehydrogenase, alkaline phosphatase can be estimated to give any indication of subclinical mastitis. Then you can do ELISA to estimate neutrophils, is still an experimental test. Then there are certain other tests like ultrasonography, and you can go for infrared thermography. So ultrasonography, you can use 5 to 8.5 megahertz probe is used. You have to place both the sagittally and transversely to the teat, and normal saline can be infused into udder to increase the contrast. So what you expect is that in inflamed areas you will get a hyperechoic hyperechoic areas of inflammation, and you can measure the supramammary lymph node, and so that again will give an idea of inflammation. So ultrasonography is another method. You can go for infrared thermography. So you take a infrared camera, take a picture, and you can estimate the temperature. But for that there should be certain environmental conditions to be maintained, like there should not be direct sunlight, there should not be wind, the udder should be washed and dry, or if there is dung pasted on the surface of the udder, then you won't be able to estimate. So it's very easy to do, and it is said to help in early detection of clinical mastitis. So this is very valuable when it comes to large number of farms where you have hundreds of cow and you are doing robotic milking and all. So if you do infrared thermography, then these cows with the mastitis can be detected fairly easily even before the milk is drawn. Then there is biopsy. Biopsy is done in research purposes, where we have to study the pathogenesis or study the changes in the epithelium, mammary epithelium and all. You have to go for biopsy. Now, what are the clinical pathology or changes in the blood when it comes to mastitis? As I told in the previous class, gram-negative bacteria, because of this endotoxin release and all, will produce a systemic sign. So there will be significant clinical changes, and you can see the clinical pathology directly in case of gram-negative bacteria. So there will be leukocytosis, PCV will increase, creatinine burn, etc. will increase because of this toxemia. Then monocyte count and lymphocyte count will get reduced because of toxemic changes. But when it comes to gram-positive cocci, the cow component will be it will be almost normal. So you won't get any significant changes when it comes to clinical pathology of mastitis due to gram-positive cocci. So that is a bird's eye view regarding diagnosis of bovine mastitis. Now we will